Hello everyone, welcome to BioZee, and today we are going to learn the concept of ploidy. So let us first understand what exactly is the meaning of this word ploidy. So ploidy is actually the number of sets of chromosome in a cell, or we can say in an organism. Now what do we understand by this set? So set simply means in a chromosome how many copy of each chromosome present. Set simply means copy of a chromosome. Okay, for example, let's say this is a cell, and in this cell we have one chromosome of this type, another chromosome of this one, and one more chromosome, let's say this one. So for every chromosome you can see just one copy is present. Since only one copy of each chromosome is present, that is why we have just one set present, and that is why the ploidy become n. How do we represent this ploidy? Ploidy is represented by n. So this condition is known as haploid condition. Likewise, let's say we have in this cell, now for every chromosome which we drew earlier, two copies present. Right? So if two copies of every chromosome present, so I can say there are two sets present for each chromosome. Right? So in that case the ploidy become 2n. And this is known as the diploid condition. In the same manner, let us take another example, another cell having now 3 copies of every chromosome. So we have 3 copies of every chromosome. That is why now I can say that I have 3 sets present for every type of chromosome. 3 sets. So 3 sets, that is why the condition is 3n, which is termed as triploid. And in the same manner we can have the tetraploid, the pentaploid, the hexaploid, all these conditions. Okay, so this is about the ploidy, number of sets of chromosome. And to understand how many sets of chromosome a cell is having, you need not to look for the total number of the chromosome, but the number of chromosome for every type. If one chromosome or one copy of every chromosome present, then ploidy becomes n. If two copies of every chromosome, then ploidy becomes diploid. Right? So this is about the ploidy. Now talking about the amount or content of DNA. Amount or content of DNA. So when you wish to look for the amount of content of DNA, just look for the chromatids, the number of chromatids present in every chromosome or every type of chromosome. Okay, and this amount of DNA is represented by C. So again, let us take few example and understand. Let's say in a cell there is this one chromosome present. How can I say this is one chromosome? Just look at the centromere. One centromere, this is the centromere region. So one centromere, if there is one centromere, so I can say this is one chromosome. But in this chromosome, how many chromatids are there? Just single chromatid, single DNA thread. That is why this is condition C. It is just C. Okay, now let us take another example. Now I have two chromosomes because two centromere we can see. So two chromosomes. And these two chromosomes contain how many chromatid? 1 and 2. So these are same type of chromosome having two chromatids. So the condition become 2C. Likewise, take another example. This and another type of chromosome, this. So now we have one chromatid for this chromosome and again one chromatid in another chromosome. So every chromosome, every different type of chromosome has just one chromatid. Again the condition becomes C. Again the condition becomes C. Let us take another example quickly. Here. Count the number of chromosomes. There are 4 centromere present, so 4 chromosomes. 4 chromosomes, but we have to look for each type. So if I look just on one, that is the white type. So white chromosome are just 2. So they have 2 chromatid. Now if I focus on the other one, the red one, again they have also 1 chromatid and 2 chromatid. So for every chromosome we have 2 copies present and they are carrying 2 chromatid. So the condition become 2C. 2C. Now let us take many more examples and understand this concept clearly. So first, just tell me this case. See, 1 centromere, so 1 chromosome. Is there any other copy present for this chromosome? No other copy of this chromosome, just 1. So the ploidy N, just 1. And how many chromatid present here? Just 1 chromatid. So C. Okay. Then second example. Let's say the condition is this now. How many centromere? 2 centromere. Are they same chromosome? Yes, same. So for 1 chromosome I have another copy present. 2 copy of 1 chromosome. So ploidy become 2N. Ploidy become 2N. And same type of chromosome are carrying one thread and another thread. One chromatid, another chromatid. So 2C. Content become 2C. Now let us take another example. I have this chromosome and I have now sister chromatid attached to this chromosome. So in this case, since 1 centromere is present, so the chromosome is just 1. Chromosome is just 1. And is there any other copy of this chromosome present? No. Chromosome copy is not present. So the condition is N. 1 centromere, so 1 chromosome. You can't say these are 2 copies of chromosome. No. These are the sister chromatid, not the chromosome. So chromosome is just 1. But this 1 chromosome is carrying how many chromatid? 1 chromatid here, other chromatid here. So content is 2C. Content is double, 2C. The ploidy is N, but content is double. Let us take another example. See in this case. How many copies of every chromosome? 1 copy of white, 1 copy of red. So 1 copy means 1 set of every chromosome. That is why the ploidy is N. Now coming to the content, white is carrying just 1 chromatid. Likewise, red is also carrying 1 chromatid. So every chromosome is carrying 1 chromatid. That is why the content is again C. Right? Let us take another example. So now we can see that white has 2 copies and red has 2 copies. So since white has two copies present, and red also has two copies present. So every chromosome had two copy present. So I can say the condition is 2n. 2n. And white is carrying one thread and two thread. So two chromatid. Likewise, red is also carrying one chromatid, two chromatid. So the content become 2C. Right? Now let us take some more examples. Let's say the condition is this now. So here again, look at the chromosome. Since one centromere here, another centromere here, so one chromosome of each type. So this chromosome, this is another chromosome. So one chromosome of each type. That is why the condition become n. There is no other copy present. So condition is n, right? But every chromosome is having how many chromatid? One chromatid, two chromatid. White has two chromatid. Red also has one chromatid, two chromatid. So the condition become the content of DNA become 2C. 2C. Okay. Then coming to another example. Let's say here we have one chromosome like this, another chromosome like this, this. See, so total number of chromosomes are 4, because you can see 4 centromere are present. So 4 chromosomes are there. But for every type, is there another copy present? No. Only one copy for every chromosome. So the ploidy remains n. One set of every type of chromosome. Another set is not present. Another pair is not present. Right? Okay. Now look at the content. White has one chromatid. Red has one chromatid. Blue has one chromatid. Yellow has one chromatid. So one chromatid in every chromosome. That is why the content remains C. Content remains C. Okay. Now we are moving towards another example. Another example let us take like this. So look at the centromere. Just one centromere present means the chromosome is just one. One chromosome. And is there any copy of this chromosome present? No. No copy present. So I can say that one copy of this chromosome. So n, ploidy is n. But how many content of DNA? How many chromatid it is carrying? One chromatid, two chromatid, three chromatid, four chromatid. So the content has become 4C. 4C. Okay. Likewise, let us take another example. This, and this. So now we have 4 chromosomes present. But can you see any other copy present of these chromosomes? No. White has just one copy. Red, one copy. Blue, one copy. Yellow, one copy. So one copy of every type of chromosome. So the ploidy remains n. One set of every type of chromosome. But how many chromatid every chromosome is carrying? So white is carrying one chromatid, two chromatid. Red is carrying one chromatid, two chromatid. Blue is carrying one chromatid and two chromatid. And again yellow is carrying one chromatid, two chromatid. So the content, every chromosome is carrying two chromatid. So the content is 2C. 2C. Okay. Now let us take one more example. What do you think what is going to happen in this case? Try to figure it out. Right? So again, how many copies of each type of chromosome? For white, we have 2 copies. For red, we have 2 copies. For blue, again 2 copies. For yellow, we have 2 copies. So for every type of chromosome, since we have 2 copies present, 2 sets present, that is why the ploidy become 2n. Now let us count the chromatids. White has 1 chromatid, 2 chromatid. Red has 1 chromatid, 2 chromatid. Blue 1 chromatid, 2 chromatid. And yellow 1 chromatid, 2 chromatid. So the ploidy become 2C. Now just one more example, last example. And I hope then the concept of ploidy would be clear to you people. Alright people, so look at this. Again, 2 chromosomes of each type, because one centromere, another centromere, right? For red also 1 centromere, another centromere to 2 chromosomes. Then for blue also 1 centromere, 2 centromere, so 2 chromosomes of blue, and likewise 2 chromosomes for yellow. So we have 2 chromosomes present for each type. The ploidy remains 2n. But when we look at the content of DNA, so for white we have 1 chromatid, 2 chromatid, 3 chromatid, 4 chromatid. Likewise we have for red 1, 2, 3 and 4 chromatids. Likewise for blue 1, 2, 3 and 4. And in the same manner for yellow 1, 2, 3 and 4. So 4 chromatid present for every chromosome. That is why the content become 4C. Alright. So in order to conclude the whole video, in order to understand the ploidy of a cell, you need to look at the copies of every type of chromosome. Copies of chromosome. And if you want to look at the content or amount of DNA, then look for the number of chromatids every chromosome carrying. Alright, so this is going to give you an idea for the concept of ploidy and the content of DNA. Hope you like the video. If you like the video, please hit the like button, share and subscribe. I will see you in the next video. Bye bye bachcha log. Take care. Keep shining.